has any papers, ideas, essays they want to present at that conference, you can contact me, or you can send a query to the email address that we mentioned there. We are super excited, and we're hoping it's a really broad interdisciplinary event. We've been doing a lot of stuff with Global Philippine Studies at NYU, and we're really hoping to build a community and celebrate and think about this scholarship, and to think about 500 years of colonialism and resistance. Cool. So I think that's about it for me. We're going to have 4 really excellent presenters today, but we're going to start with our opening speaker, and Noelle, I think you're going to introduce her. Oh, just very quickly, when you have we want everyone to feel free to ask questions, and so when you have questions for participants, for the presenters, go ahead and throw them in the chat, and we will ask them. So each presenter is going to have a few minutes to speak, and then we'll have a couple minutes of question and answers with each presenter, and then we're going to at the end, we're going to have kind of a discussion with everybody. So Noelle, do you want to go ahead and introduce Yasmin, our opening speaker?
Yep. Is there a digital version we can share of your conference? That's a question to you, Michael. So yeah, okay, but we'll answer that later. And I'm super happy and excited to introduce our moderator. Yasmin Tayag is a science editor and journalist, currently working as the lead editor at the science website Future Human, and as a reporter doing daily coverage of COVID-19 at the Medium Coronavirus blog. So she received her master's in biology at NYU, where she did developmental genetics research on the formation of the early heart in the model organism, Siona intestinalis, whatever that is. Aside from that, Yasmin's a great cook, a great friend, and a dog mom to 8-year-old Buko. So take it away, Yasmin.
Thank you, Noelle. Sorry for throwing Siona intestinalis at you, but you got it perfectly. Hi, everyone. Thank you, Noelle and Michael, for having me, and welcome everyone to the science-themed edition of the Filipinx PowerPoint Party. As Noelle mentioned, I'm not currently a scientist. I work as a science journalist at the science website Future Human and at the Medium Coronavirus blog. I'm excited to be here today for a number of reasons. When I was at NYU doing my master's way back in 2009, I had no idea what I was doing. I had just moved to New York from Toronto, where I had lived all my life in a very tight-knit Filipinx community. I think I saw in the chat there's someone from Toronto here, so hey, you know what it's like. Tight-knit Filipino community, great. My parents didn't know anything about academia. I didn't know, nobody told me I was supposed to join a lab, to write a thesis, to teach undergrads. To me, I just thought grad school was a continuation of undergrad. I figured it out on my own eventually, but I felt jealous of all the students whose families, walked them through every step of the way to study advanced science because, they had done it here too. And at NYU biology, I didn't meet any Filipinx people. It was a big change for me. I felt lost in the lab, and I wanted so badly to see someone who looked like me, just for support, for a familiar face, someone to make me feel like, yes, I should be here too. So looking at all of the incredible scientists who are here today, I feel so encouraged that others who follow in our footsteps aren't going to feel as isolated and as out of place as I did. So fast forward to now, obviously not a scientist anymore. And now in my work as a science journalist, I write and assign stories about science and how it's being used to address the world's problems. I run into similar issues of representation in my field too. there is so much research from old white guys and not enough from the diverse people who make up this planet, people like you and me. And that's important because the biggest issues we are facing as a planet right now, climate change, extreme events caused by climate change like typhoons and floods, pandemics, they are often felt most keenly and often disproportionately so by people of color around the world, people like our families here and back home. But people like the scientists here today are changing all of that. You hear people say representation matters, and it does, especially in science, because it has an impact on the science that is pursued, the science that gets funding, and the people that that science will impact. So today you'll hear from Ingrid Paredes, who is studying clean energy technology, which will be absolutely critical if we want to get climate change under control. Similarly, JP Balmonte's work studying diminishing Arctic sea ice is also critical for the study of the climate. Carla Bertulfo is deploying the very powerful genetic engineering tool CRISPR, shout out genetics, to find new ways to treat cancer. And finally, Kathleen Gutierrez will take us back to our literal roots by teaching us to view Philippine history through the lens of its native plants, giving us a new perspective on our nation's ancient history. Needless to say, I'm so excited that these people are representing the Philippines in the sciences, and I am extra excited to hear from them today. So thank you and enjoy the event.
Fantastic. Thank you, Yasmin. We are so excited for this event, and I am so excited to introduce our first speaker, who is Dr. Kathleen Cruz Gutierrez. Dr. Gutierrez is an assistant professor in the Department of History at the University of California, Santa Cruz, where she teaches classes on the Philippines and modern Southeast Asia. She completed her PhD in Southeast Asian Studies with an emphasis on science and technology at the University of California, Berkeley. Her research explores colonial botany, agentic plant studies, and textile weaving technologies. With Paul Michael Atienza, who I believe is here. She is guest co-editing a science and technologies studies themed special issue of Philippine Studies, Historical and Ethnographic Viewpoints. And in 2021, she'll be in residence at the Humanities Institute of the New York Botanical Garden as a Mellon postdoctoral fellow to complete her book manuscript, tentatively titled Sovereign Vernaculars, Philippine Botany and the Dawn of New Imperial Science. And so we are so excited to hear from Dr. Gutierrez and take it away.
Yes, oh, thank you so much, Michael, for the introduction. Yasmin, of course, for introducing the panel. And it's so good to have, yes, my co-editor here as well, Paul Michael Atienza. Keep an eye out for that special issue. I'm gonna go ahead and share my screen now. All right, I think that's Michael, just a thumbs up. Is that looking okay? Yes. So I should again first thank Michael and Noel for giving me such an exciting venue to share a little bit about my work. I'm grateful for that science takeover theme for today's PowerPoint party. Y'all have allocated some time to us humanists intent on charting the history of science. At the same time, us humanists also try to probe the positivist investment in science so that we may better assess the imperial, colonial, and post-colonial dynamics embedded within science and frame science as one of the many more domains of knowledge with its own culture, practices, belief systems, and virtues. In the next 6 or so minutes, we're going to blast through eras of the Philippine past through the lens of botany and the role of botanists and plants in this spectacular field in which I consider my home discipline. The plant sciences and really botany, which is my area of expertise, demonstrate how intellectuals have worked to assert the Philippines' floristic place in the world, thereby advancing its territorial existence as defined by plants. Here's an example of one such Philippine botanist and artist. His name is Regino Garcia. I'm not going to cover him in this presentation, but I share him with you here to incite some curiosity into this history. I'm going to close this presentation with a reflection on something I've been chewing through lately that I'm calling the agentic role of flora in Philippine history, so that we might begin to think with plants as we write the past. But before all that, I should say a little bit about myself. So I'm very proudly a product of California public schools, K through PhD. I'm trained as an area specialist of Southeast Asia, and I really got here by way of public health. So prior to my academic career, I worked in public health and identified the history of public health as my possible inroad in for my doctoral research. And medicinal plants hold a central role in Philippine public health and were my way of getting into the plant sciences. This is an image of the bayabas or guava, which has been promoted by the Philippine state to have excellent medicinal properties. I soon discovered in my earliest research how plants were discursively positioned to reify the nation state form, most especially following World War II. Books, for instance, on medicinal plants from the Philippines emerged to highlight traditions that uplifted the spatial idea of the newly sovereign Philippines. And so even if this bayabas, for instance, came to the Philippines by way of the Spanish colonial galleon trade, the state now could assert the plant as its own, as part of the healing environment that the Philippines could offer. Now I find this interesting because of the kinds of matrices of meaning imbued in plant life, when, as we know, plants don't have the same loyalties that political projects may pursue. And so this has gotten me to think about all the ways in which people and institutions try to lay claim to plant life. So I decided to write a dissertation that looked at colonial botany under both the Spanish and US colonial regimes during a moment when a new internationalist science emerged at the turn of the century. And I argue that new science worked to place the Philippine colony in regional floristic formations, not just colonial ones, that attempted to cohere landscapes of the Philippines with that of the Dutch East Indies, British Malaya, and even French colonial Indochina. I'm not going to get into the details of scientific regionalism here, although I'm very happy to talk about it during the Q&A. But I will say that that project launched me in a totally different direction, where I'm now wanting to peel away from strict reliance on colonial archives in order to center the role of local plant nomenclature and local botanical knowledge in Philippine history at the turn of the century. And Yasmin sort of pointed to this during the introduction. And so for this newer project, I look at the varied local nomenclatural practices, for instance, surrounding rice varieties, the plant-based tools in textile weaving and dyeing, and even this, the sampaguita. So as many of you might know, the sampaguita is the national flower of the archipelago. It's a non-native species of jasmine. It's quite well known, especially in popular culture. It's inflected with many prototypical feminine traits, especially because of its whiteness, its aroma, its softness. And based on scientific and cultural production at the end of the 19th century, I actually argue that Manila-based elites mapped the sampaguita onto an entire archipelago to conjure national territoriality through a gendered and gender-making sensory experience of a single flower. I show how a flower's gendered and gendering characteristics were originary to the idea of the reproductive woman as the exemplar of the Philippines. Now, historian Raquel Reyes has written an excellent book on gender in the moment of 19th century national formation, but I believe that there's a place for plants in this discourse too, so that we can continue to problematize national formation and the way that gender played a role in it. So now that I've talked a whole lot about humans ascribing meaning onto plants, I'm going to flip the script for a little bit, just for a short while, to leave you with something that to think through and maybe think with me. And this is part of a separate project that I'm calling the Cold War Philippines in 5 Plants. So this is the Imelda lily. Welcome to her. This is a toad lily, called such because of its distinctive markings, or these purple spots. Now, the species was discovered during an early 1970s expedition to the Tasaday, an alleged Stone Age cave people discovered in southern Mindanao during the presidency of Ferdinand Marcos. Now, this lily was named in honor of Imelda Marcos. News articles celebrated the discovery, lauded the plant's uniqueness, and allegedly printed the lily on stamps to give further esteem to the First Lady. Now, as some of us might remember, the Tasaday were determined to be a hoax, likely contrived by the administration to detract attention from the mining campaigns in Cotabato and the declaration of martial law in 1972. Eventually, the Imelda lily became the butt of jokes. Since locals use the sap from the flowers, you crush it, right? You crush them, you can catch slippery amphibians. They used it for catching frogs. Critics use the analogy to pit one toad lily against another toad lily. And now this lily, as it turns out, was actually first discovered in Taiwan and is only understood to grow in Taiwan. So botanists had to re-identify it as the Triceratops formosana, which it had been originally named, reducing the Triceratops imeldi after Imelda as a synonym. But this begs the question, how did this lily jump an entire sea and an archipelago and wind up in Mindanao among the Tasaday? Now we can postulate the reasons for why this might have happened, but I pose this possibility, that perhaps the lily, existing well outside its supposed growing range, hoaxed the hoaxsters of the fantasia that was the Marcos regime. And so with that, maraming salamat everyone. I thank you so much, and I leave you with the first botanist that I ever came to love. It's my father, a specialist of the Philippine Dipterocarpaceae, who remains a critical interlocutor and inspiration behind my own work. Thank you again.
Thank you. This is fantastic. I have so many questions. I want to encourage everyone to feel free to throw your comments in the chat. But I wanted to start off, Dr. Gutierrez, by asking, when we think about plants, you mentioned agentic structure and plants having agency. And so I was wondering if you could elaborate more on how you see that concept operating in your work.
Yeah, so I'm trying to really play with it when it comes to historical writing. And I'm not really trying to push the idea that plants as agents are making sentient decisions to run human history, although how cool would that be, right? I think we're kind of moving in some direction with that, some research. But I am trying to show plants as sort of collaborators in some way in the unfolding of human history. And I think when it comes to the Imelda lily, you have this supposedly misplaced plant that's growing outside of Taiwan that just sets off a flurry of intellectual and political activity. And there's something really fascinating there when we're in fact, maybe as humans, really at the helm of unique plant characteristics, or even when plants are operating a little bit more strangely. And so in this sense, historically speaking, plants become a bit more collaborative. And I think that this is really also tying into maybe more of the interspecies dialogue and discourse we're seeing in other disciplines as well. But hopefully that kind of gives a little bit of a sense of what I'm aiming towards, Michael.
Absolutely, absolutely. Alex has a question. Alex is thinking about this question of flowers as the kind of representatives of the nation. And so what do you think about a non-native flower being the national flower of the Philippines? And do you have any other ideas for what that might be to avoid Manila centrism?
Oof. Yeah, I'm going to try to prevent the pitchforks at my door for saying anything bad about Sampaguita. It's all good. It's all good. But I will say this. The fact that it's a non-native species actually does not press as much concern onto me. I think that this kind of question of nativeness becomes a little bit dangerous. And it actually smacks of a lot of the nativeness discourse that can happen, for instance, with populations that move across borders that are actually quite new. But the problem that does arise, though, is yes, what Alex is pointing to, this kind of Manila-based idea of what exactly a national flower should be. As recently as 2012, there's been this push to actually promote a different plant, an orchid from Mindanao, as one of the national flowers of the Philippines. And I think the discourse around this is fascinating too, because there's so much debate about what this might mean by allowing Sampaguita to share its mantle with a different flower. I argue that a lot of the discomfort around why people don't want to elevate this orchid is not only because of maybe the regional hesitancy around bringing up a plant from Mindanao, which, as we know, has historically been folded outside of sort of imperial Manila formations of the state. But actually, it's a question of gender, and that the Sampaguita has such a long-going association with the prototypical Philippine feminine that if we actually introduce another flower that does not have that heritage, we might actually be disrupting the idea of that ideal feminine altogether, and maybe open up the possibility for new formations of it too. So I'm not completely against bringing in a new flower. I'm just very curious about where the debates are going to be headed.
Lowell has a question. Lowell wants to know, how does our understanding and developing of botany as a science differ between Western explorers and those of native-born peoples? I know there's a lot of aspects to this, but if you have thoughts.
Yeah. Thank you so much for the question, Lowell. I love this question because I actually think it helped put me in a very different position even when I was starting out my research. One of the communities that I write for and I dedicate my scholarship to are Philippine botanists, especially those who are prosecuting work in the Philippines. And I remember kind of presenting something where I had used the term settler colonial science and put botany in that realm. And I had botanists confront me and say, well, if there's nothing settler colonial about this, so what's going on? And I think that made me recognize, one, the kind of bias I come in as a US-based academic, and two, the kind of sensitivity I need to deploy for how science has changed over time, especially in the post-colonial period. There are intellectual genealogies that are absolutely traceable to both the Spanish and US colonial periods, but this does not discount the fact that there is science that is growing and changing in the Philippines as well, intent on advancing intellectual knowledge in ways that are divorced in many respects also from that maybe more violent history. And so I think there is a difference between the botany of exploration from like the early modern period that we might see in early modern Philippine history and the science that we see today in 2021. And what I love though is looking for that specificity, right? It's really hard to sort of blanket a generalization that differs or separates the two, but understanding where the historical contingency comes in and changes that scientific practice.
Tremendous. Everyone, let's give a big round of applause for Dr. Gutierrez. That's fantastic. So fascinating and so interesting to think about, you know, obviously the historical context, right, of all of this science and all the research that we're going to be discussing today. Noelle, do you want to start with our next presenter?
Yes, thank you, Dr. Gutierrez. That was fascinating. I'm very excited to introduce our next speaker. Our next speaker is Carla Bertulfo. She's a PhD candidate in biological sciences at Columbia University. Her research utilizes high throughput genomic technologies to discover biomarkers and combination therapies that could improve T-cell leukemia patient prognosis. So very simple stuff. Outside of the lab, Carla is very passionate about her work as a community organizer, fighting against social injustices in the Philippines and in the US. She's also the founder and chairperson of the Association of Filipino Scientists in America, an organization born out of the recognition of the important role scientists play in serving our communities. We reached out to Carla to organize this whole event. And so really big thanks to her for collaborating with us and also for presenting. So without further ado, take it away, Carla.
Thank you, Noelle, and thank you, Michael, as well, for really reaching out to AFSA for this collaboration. I'm really excited to share my research as well as the advocacy that I've been doing together with AFSA. So I actually work at, so my research is on T-cell leukemia. That's where the Ferrando Lab at the Irving Cancer Research Center in Colombia has been focusing on. And T-cell leukemia is really a disease or a blood cancer characterized by the presence or crowding of immature T cells in your blood, bone marrow, and lymphoid tissues. Patients with T-cell leukemia really experience fatigue, weakness, and they're very prone to infection. T-cell leukemia is still a disease to beat. It's aggressive, it progresses quickly, it's resistant to therapy, and its secret weapon is really the mutations. So mutations are permanent genetic alterations in your DNA that could confer some cells to be cancerous. And one of those mutations in T cell leukemia is the Notch1 mutation. Notch1 mutations really work by directing genes to express abnormally in cancer. And so a lot of attention has been really given into how do we target Notch1 in T cell leukemia to improve patient survival. And one of those drugs that has been developed is the drug called DBC, which is a gamma secretase inhibitor. And this really works by preventing Notch1 from getting into contact with the genes that it wants to abnormally express. In my experiments, I've seen that DBC is really effective against T cell leukemia. And I've seen this in T cell leukemia lines that I've treated with DBC. The cells are actually shrinking when you treat them with DBC. So it is effective, but not quite. Because in the same experiment that I did, there's also decrease in the cell count, but you see that DBC treated cells do not completely die, or not all of them actually respond to DBC treatment. So this is the central question of my research, and that is really how do we improve patient response to DBC? And there are two corners in this drug discovery arena. In the red side corner is really to ask what genes allow the leukemia cells to resist DBC treatment, and we call them the resistance genes. And the blue corner to ask what genes can we additionally target to knock out the leukemia cells, and we call them as sensitivity genes. To answer these questions, I've used a very cool genome editing tool called CRISPR-Cas9, which is something that is adapted from the bacteria. It is a challenger in the field because it is very efficient, easy to use, and cheap, and it works by deleting or knocking out a target gene. And the secret weapon of this challenger is that it can be used to knock out thousands of genes simultaneously, and I'm going to talk about that later. So how does CRISPR-Cas9 work? So if you have a target gene, you basically infect your cells with a guide RNA that locates the target gene. And this guide RNA will guide a Cas9 nuclease protein into the target gene to induce cuts in your DNA. Your body has a normal response of putting and gluing all these DNA fragments in your body and repairing them. But once it's repaired, you actually take out the missing pieces, and now this gene is considered non-functional. And this is what we call as gene knockout. This is a really cool technique, and what's more exciting is that several researchers in leading institutions has been able to elevate this technology such that now you could target thousands of genes in the human genome simultaneously in one experiment. And this is what I'm doing in my research work, where I do genome-wide CRISPR-Cas9 screening in T cell leukemia cells, and I target 20,000 genes in the human genome. And how is it being done? Well, the guide RNAs are actually pooled in a viral vector and these are hundreds and thousands of guide RNAs that you infect into Cas9 expressing cells, and you treat them with the drugs. You sequence them through high throughput sequencing, which is kind of like robotics analysis. And you do a bioinformatics analysis to check the abundance of guide RNA to know which genes are responsible for resistance and sensitivity to the drug. So I've done this in CRISPR-Cas9 in DBC treated T cell leukemia, and I did get statistically significant results for this experiment, although the names of the genes I cannot divulge yet because this is still an unpublished data. But there's one gene that I want to really highlight here. And this is a gene that our lab has actually already found out a decade ago, where we published in Nature Medicine that the loss of this gene actually induces resistance to Notch1 inhibition in T cell leukemia. So it kind of validates my experiment and got me really excited into looking at these genes, because there's a lot of opportunity involved with this one. Because one, you could really discover biomarkers that could predict response to therapy, so that if you have a gene that you know could confer resistance to DBC treatment, you will take a step back and not treat your patients with DBC because you know they will be resistant. Another approach that we could really take is to develop combinatorial therapy to improve patient survival. So that when you know that gene X actually confers sensitivity to DBC treatment, you develop drug Y that could actually target gene X so that you will knock out your cancer cell and ultimately improve patient survival. So that's the gist of my research. I know there's a lot of technical details, but I'm very open to answering your questions. But I do want to acknowledge the lab that I've been working with. We've been knocking out cancer, but this is not an individual work. It is actually a collective work of the community. The same actually with knocking out the cancer that plagues in our society. It is always a work that is collective, and you need to be a part of the community. That's why outside of the lab, I'm very active with different social justice organizations. I'm actually part of the National Democratic Movement for the Philippines, fighting for true independence and democracy. And you can see here some of the organizations that have been really involved with that causes, and you might want to check them out. We also have different US chapters. But really, that's also the reason why I founded AFSA, because I see how important the role of scientists are in learning and serving our communities. It's time to really go outside our laboratories and also, play our role in being catalysts of change, and to learn from our communities and to speak out and use our platform to impact change. So some of the advocacies that we have in AFSA is really the endorsement of the Philippine Human Rights Act. That it's a bill that we want to propose with the US Congress. We have been endorsing this, and it was led by a lot of different human rights organizations with the objective of really suspend the US military aid to the Philippines, because that money, I guess they've given $500 million to the Philippines in the past years, has been being used to really fund the different human rights violations in the Philippines. And there are laws that try to really be weaponized against the people. People expressing dissent or being tagged as terrorist, really. And AFSA also has a Filipino healthcare workers campaign that really seeks into looking at the rights and welfare of the Filipino healthcare workers that were disproportionately affected by the COVID-19 pandemic. And with that, I'll be happy to take any questions that people have. Thank you. And please reach out to AFSA if you have anything.
Thank you, Carla. We have a few questions, or one question, and then I also have one. And we'll start with Fred's question. Oh, by the way, I love the graphics. I feel like that was super effective. I never felt like we were lost. And so
Thank you.
So Fred is asking, do you plan to do a mouse knockout?
Yes, knockout, right. Yay.
For their high throughput screen to show in vivo functionality?
Yes, that will come up later in the experiments. My plan really is to functionally validate each genes. And I'll be doing them in cell lines first, in T-cell leukemia cells, before I actually move on into mouse models. But our lab do mouse work as well to confirm this data.
And then a question I had is, how do you even begin to narrow what gene you'll look at to see what confers resistance against DBZ? Is that like cast a wide net, fish for the gene, or like so like that whole process?
There's really a lot of processes, like a lot of ways you could go into it. But for now, what I've been doing is really do another CRISPR screen with another T-cell leukemia line. And what I want to do is cross-check the results of both experiments and see like which genes are consistent between two systems, so that I would know that this gene is generally applicable for different T-cell leukemia background. That's one approach. And another approach is really like looking into each gene one by one and looking at the literature if they've been implicated in T-cell leukemia development. And there are actually some genes that are really interesting, but I'm also like just working on the other experiment just to save time with this functional validation.
That's super cool. And I know you can't divulge the identity of the genes. So from the first moment you study them to like publication, how long is that process usually?
I think it takes my whole PhD. I would suppose it's like 3-4 years. Well, the whole PhD takes 5 years, but I'm hoping like it will take me 3 to 4 years to finish, including like the functional validations and like the mouse work. And hopefully we could like translate it in the clinic where we could actually have preclinical models when we develop drug Y, for example. Did you collect any? Sorry, I've been asking the. I've been looking at the chat.
That's the question. Yep, that's
okay.
You collect.
Right now in my CRISPR screen, no, but I don't know like which part of the experiment is this question being asked, but in my CRISPR screen experiment, I infect the cells using a green fluorescent protein marker, and that's what I do to indicate that these genes were infected with the guide RNA, and I use flow cytometry for that.
All right. Okay, so for people who have questions, we have time at the end, and I definitely had questions about the science advocacy part. But moving on for the sake of time, I'll pass it over to Michael to introduce the next speaker. And thank you, Carla.
Thank you.
Absolutely. I'm going to just one more time, really quick, share the info for our virtual tip jar. A couple people have already donated. We really appreciate it. But if you want to help support the advocacy work that Carla and our friends over at AFSA are doing, as well as the incredible climate justice work that you'll hear about when Ingrid presents. So thanks to those who have already donated, and then we appreciate your support. I am so excited to introduce our next speaker, who is coming to us all the way from Denmark. So thank you, John Paul, for staying up late to let us hear about your research. Dr. John Paul Balmonte is a postdoctoral researcher at the University of Southern Denmark in the Center for Hadal Trench Research and the Nordic Center for Earth and Evolution. His research tries to understand the role of bacterial communities in transforming compounds in oceans, rivers, and lakes, which has implications on the global carbon cycle, climate, and food webs. He advocates for storytelling as a way to make scientific information widely accessible to the public, and to help lessen the information gap between those who conduct the research and those who pay for it through taxes, as well as to combat the spread of information. So Dr. John Paul Balmonte, thank you for taking the time to reduce the information gap for us, and take it away.
Thank you. That is combating the spread of misinformation. We want to spread information. No worries. Okay, let me go ahead and share my screen. Great. Can you all see my screen? Good. So, all right. So, hi everyone, and thank you so much, Noelle, Michael, and Carla, for the invitation to give this talk. This talk is about this expedition where I participated as a scientist, and it was a big international Arctic drift expedition headed by Germany, but with 20 other countries. And I have here the title of my talk is The Largest Arctic Expedition to Understand the Diminishing Sea Ice. And this expedition is named Mosaic, and it stands for Multidisciplinary Drifting Observatory for the Study of Arctic Climate. And this expedition actually received quite a bit of attention from the media, in part because of its very timely goals of understanding how the things that are happening in the Arctic affects the rest of the world, as Yasmin talked to us about earlier. What happens in one place really doesn't stay in one place. It affects the rest of the world. So just to get us situated here, I don't know how many of us are familiar with the Arctic, but this is a bird's eye view of the Arctic, and the red dot and the red arrow indicate the North Pole, the geographic North Pole. The Arctic is in the north. It is a home of polar bears, snow penguins. And just as a point of reference, most of you are probably in the United States, and some I saw in Canada. And that's here in the lower left corner of the map. And I'm right here in Denmark, as indicated by the blue arrow. And so why conduct Arctic research? Why pour so much money and resources and time and effort into doing this Arctic research? Well, in the past few decades, the National Snow and Ice Data Coverage has been tracking the sea ice extent in the Arctic, and that's essentially the surface area of sea ice in the Arctic. And what they've noticed is that since 1978 all the way to last year is that there's this very, very steady decline of sea ice surface area or sea ice extent. And they actually project that this downward trend will continue in the next few decades, such that it's very possible that the Arctic is going to be seasonally ice-free. And that's a big change in paradigm, because what we thought was a permanent feature of the Arctic, the sea ice, can actually vanish in the summer. What exactly are the implications of a diminishing sea ice? So like Carla, I have these schematics to simplify the concept. And so essentially what we have, or what I've drawn out here, is a cartoon depiction of the water column. So the ocean, the Arctic Ocean in its past and current form, still with sea ice on top. And to the right, we have an Arctic Ocean that is in the more depicting present and future scenarios, and that's with a diminishing, thinning and breaking sea ice. And as we move forward into the future, there are some things that are likely going to happen to the Arctic Ocean. So the diminishing sea ice is going to make way for a lot more light to enter the water column, and this sunlight is going to warm up that water. And what that does together is that it stratifies the water column such that there's warm water sitting on top of cold water. And that's a bit of a concern, because actually a lot of the nutrients that's powering the organisms and the reaction in the upper part of the water column comes from the deep ocean. And when the water column is stratified, warm water, cold water, it actually prohibits a lot of this vertical mixing that introduces a lot of deep water nutrients back to the surface water where nutrient is relatively depleted. Now, why should we care about these physical conditions and the chemistry here? Well, I'll bring us back to an equation that maybe some of us have seen in high school and college. But essentially this reaction over here in the middle of the slide tells us that carbon dioxide plus water, with the help of some energy from sunlight, is converted to some type of organic matter and oxygen. And as you guessed it, this is photosynthesis. Plants carry this out on land. In the ocean, they tend to be macroalgae, algae, or phytoplankton, very, very microscopic organisms. The reverse equation can also happen, and this is called respiration. So this is the conversion of this fixed organic carbon in this organic matter by use of respiring oxygen and turning this carbon that is organic into inorganic carbon, carbon dioxide plus water. And this equation matters so much for the study of the Arctic, because this equation is what determines the balance of carbon in the system. oops. So for example, at present, scientists tend to think of the Arctic as a net carbon sink. And that essentially means that there's a greater influx of carbon dioxide that's being fixed into organic matter in the water column and less emission of carbon dioxide back into the atmosphere. And the worry here is that in the future, with the thinning sea ice, the increasing temperatures, is that there's actually going to be a tip in this balance such that respiration might actually, in some parts of the Arctic, overtake photosynthesis. So there's going to be more carbon dioxide emitted to the atmosphere than there will be fixed in the water column through photosynthesis. So the Arctic becomes a net carbon source, and that is the issue because global climate change, as we know, stems from an increase in global carbon dioxide concentrations in the atmosphere. And it's this interest in understanding these fluxes, these arrows, and the movement of different compounds and gases that really drives the focus and the goals of Mosaic. So the quote here is, "The focus of Mosaic lies on direct in situ or on-site observations of the climate processes that couple the atmosphere, the ocean, sea ice, biogeochemistry, and ecosystem." And what that essentially means is that in order to understand an ecosystem and the processes that are happening, one must look at the physics, the chemistry, the biology, and how all of those different factors are interwoven. They're very, very tightly linked, and you can only understand an ecosystem by understanding all of those facets simultaneously. So from a logistical point of view, there are a lot of challenges in trying to carry out this kind of expedition. So it's an expedition for 1 full year in which they planned to bring this German icebreaker, the Polar Stern or the Polar Star. It has ice-breaking capabilities and bring it to the Siberian portion of the Arctic, which is depicted right here, freeze it in to the ice pack for 1 full year and have it drift across the Arctic all the way back to Europe. And of course, this plan requires quite a bit of resources, and eventually fuel and food and other supplies are going to be depleted. So there must be a way to resupply and also to exchange the scientists, because there's only so much you can do when you're out there and people tire out. So they wanted to exchange scientists every 2 months. So in the end, this was a cruise track of this expedition, which began in Tromsø, Norway in September 2019 and headed up to the Siberian portion of the Arctic. These are the 4 exchange sites that are depicted in the yellow dot. And it finished last year, actually, in September 2020, and that's when the Polar Stern, the research vessel, came back to Bremerhaven, Germany. Now, here are some photos of the practical aspects of carrying out this research on ice. To the left is this graphical depiction of what the organizers and the leaders of the expedition envisioned this ice camp to look like. So there would be the research vessel and then multiple camps. So the Met City doing atmospheric work, Ocean City doing measurements in the water column, the ROV City where they would be deploying ROVs or remotely operated vehicles. And all of this requires a lot of human power because there's no power on ice. Everything, all the cables have to be set up on ice, and then they then have to be brought back, connected to the ship so that the ship can then power all of the equipment that are being used out on ice. And any equipment that is deployed to measure anything in the water has to go through the ice, and people have to manually cut these holes in order to deploy their equipment. But once all of that happened, then everyday life happened, and different kinds of sample collection on ice measurement and experiments could be carried out. So I have here a photo of the atmospheric team with their gas flux chamber measuring different gases. I have here a photo of my teammate, Celia, and she's looking at, through the microscope, some copepods. Here is Ben looking, looking at this ROV, this remotely operated vehicle, making sure that its deployment is safe. Here I am working in the dark, actually carrying out experiments with stable isotopes to look at microbial role in the cycling of different elements. Here's Jonathan looking at, preparing his drone for atmospheric measurements. And here's Amelia taking water samples from the CTD, which was brought down to the bottom of the ocean to collect water samples. And so I leave you with this image of the entire team of Mosaic Leg 4. We were in the 4th section of this expedition, and a few photos of the wildlife, copepods, starfish, and polar bears that we encountered. So I'm really glad that I had the opportunity to give this talk. And Yasmin, I think you hit it right on when you said what happens in one place affects some of the most underserved communities in the planet. So it's really important to understand the dynamics in the Arctic. Thanks. And I'll take questions.
Thank you, Dr. Balmonte. Everyone, round of applause. I have a ton of questions about what life is like on an expedition like that, and I'm sure everyone else does too. But I think I wanted to throw out just one question, which is, I know that, in the public discussions about climate change, one of the images that we think about the Arctic melting ice. And I was wondering if you had any thoughts about kind of popular misconceptions that the public might have about the Arctic, and how, you can use your research to think about that.
Sure, absolutely. I think actually one of the biggest misconceptions that I've heard so far is that, or maybe misinformation, is that there's, I don't think people quite understand that the Arctic is ocean, right? And the sea ice sits on top of water, which is very, very interesting and important from a carbon flux perspective. So, I think that people are used to thinking about Antarctica and how the ice there sits on top of land, but it's very different in the Arctic. So I think that's one of the things that I can think about. And the other thing is, as I kept mentioning over and over, I think a lot of people don't necessarily think about what happens in one part of the world and how the carbon dynamics there, the balance of carbon dioxide and organic carbon, and the role of the ocean as a carbon source or a carbon sink really affects other people globally. Atmospheric circulation occurs, and that's, from that perspective, it's very easy to move things around globally. So yeah, so some of those are some of the misconceptions that I can talk about. Yeah.
Fantastic. We have some questions here. A lot of technical stuff, so apologize if I start spreading misinformation. Do we have a historical time series for the source versus sink of sea in the Arctic over the period you showed us to see if respiration has already increased, or will that require further expeditions?
Dan, that's a great question. And my answer is there are data from other parts of the Arctic, but where we're lagging a lot of data is actually from the end of fall to the beginning of spring, right? So expeditions don't tend to go out to the Arctic in the middle of the winter or in the beginning of spring, because the sea ice is so thick that we don't have the capability to break ice over there. And of course, it's very logistically challenging, but there are data sets, there are researchers who try to understand the balance of the Arctic being a carbon source or a carbon sink. And I can actually personally send that literature to you, Dan, if you would like.
Awesome. Yasmin wants to know, do you expect the carbon storing ability of the Arctic Ocean to change as the amount of carbon in the atmosphere continues to increase?
Ooh, that's a great question. There are two components here. One is the ability of the water column to take in carbon dioxide, And I'm not a gas person, but my guess is that its capacity decreases as the water temperature increases. So I think that's one issue. There tends to be this balance, and that's one of the really important things that the ocean does is it mediates this balance between carbon dioxide in the atmosphere and in the surface waters. So when it increases in one place, it tends to then diffuse into the other. And so CO2, assuming that all the other parameters are present and nutrients that are necessary for plants or the photosynthetic organisms are present, nutrients, light, and they're not temperature limited, other trace metals. CO2 could stimulate more production, but it's actually a much more complex thing than just that because as we saw in that equation, once photosynthesis occurs and organic matter is made, things immediately get respired and done by really quick heterotrophic microorganisms whose activities and enzymatic rates actually will increase also with increasing temperature. So yeah, so those are some of the things that could happen with continued increased concentrations of carbon dioxide in the atmosphere, and actually is the reason why this topic of ocean acidification has been in the news, in the past decade and a half or maybe even longer. And that's because there is this balance. And as you increase CO2 concentrations in the atmosphere, the ocean is there to take it up. And when you take in more CO2, it acidifies the water, pH decreases, and that's another set of issues for calcifying organisms, including corals. So yeah.
Fantastic. Everyone, another round of applause for JP.
Thank you.
Really fantastic. And we're going to have, at the end, we're going to have a kind of discussion with everyone. So if anyone has more questions for Dr. Balmonte, feel free to save them for the end. But now I'm going to turn it back to Noelle to introduce our Ingrid, our final presenter.
All right, everyone. Hope everyone is staying engaged and keeping their questions ready for the discussions afterwards. Our last and certainly not the least speaker is Ingrid J. Paredes. She is a PhD candidate in chemical engineering at NYU Tandon School of Engineering in Brooklyn, New York. The focus of her research is the development of novel nanomaterial systems for clean energy technologies. Passionate about science advocacy and education, she dedicates her time outside of the lab to developing programming and partnerships as the co-chair of March for Science NYC Satellite. She's here to talk about building a sustainable future through science and advocacy. So over to you, Ingrid.
Thanks, Noelle, and thanks everyone for being here. And again, to the organizers for arranging this event. I've been learning a lot just listening to everyone speak so far. I'm gonna just turn on my screen share. Could I get a thumbs up from Michael or Noelle? Is everything good? Awesome, sounds good. And this is in present mode, correct? Not my notes mode. Amazing. Okay, cool. So yeah, thanks again for everyone for being here. As Noelle mentioned, I'm going to be speaking to you today about some of my work in nanotechnology for sustainable energy systems and how that ties into the advocacy work that March for Science New York City has been doing around climate justice. So first, I want to define what we mean by sustainable. I think this has become a pretty trendy topic for everyone from like activists to corporations to really take on. I think the UN's definition really ties it in well though. And it states simply that sustainable development meets the needs of the present without compromising the ability of future generations to meet their own need. And what this means for me as an engineer is to think about science simply as a tool for justice. I think there's been a running theme too that we've been seeing, whether we're talking about decolonizing our knowledge, to looking at cancer therapies, to looking how storytelling really impacts the work that we choose to do throughout our work. So that's kind of the lens that I'm trying to look at things at. Of course, as an engineer, though, they're kind of like two separate worlds, at least in the lab. But engineers have been thinking about sustainability, at least when it comes to our design principles. And this really comes down to three things that are all tied into our materials life cycle. So first, what are the materials that we choose to research and develop? What are the compounds that we are looking at? What does it take to actually process them, synthesize them in the lab, and then scale them up to consumer products? And finally, where do they end up? Yasmin, again, that quote that you had said in the beginning about these impacts of severe crises, usually coming on to underserved communities, is really true and perpetuated a lot, particularly by chemical industries, which I'll touch on a bit more. Which brings me to my next slide, which we're going to focus just today on a small piece of the puzzle, which is energy consumption. And maybe I misled it there. It's not really small because that is what's fueling the most, our existential crisis, which is the climate crisis that we're seeing, which is being fueled by oil and gas companies. That said, they are the major producers of the energy sources that we use today. Regardless of where the energy is coming from, just so we have a scale of like where our energy landscape is going. When we have our energy source, whether it's like oil or renewables, typically what happens is we have to convert that right directly from source to electricity, and that's done by a heat engine. So this can be like done in our power plants, or more recently, semiconductors have taken over this work. And what we get is energy out that's then converted to the electricity that we use. So the passive systems, like the front end usage of energy systems. Unfortunately, even though our lights are on, this presentation is going, what actually happens is every time this cycle happens, we lose 60% of the energy input that we put in to waste heat. So generating energy, generating electricity itself is actually a inherently wasteful process, and that's just it's thermodynamics. It's just the way that things naturally are. That said, researchers have been looking, including myself, in ways to address this issue. We can, ideally wean off fossil fuels completely and look at developing sustainable materials. We can recycle waste heat, so try to find ways to capture that waste heat and bring it back into energy cycles, which I'll touch on a bit with my work with nanotech. Or we can look at how we can increase the efficiency of these processes to begin with. And today I'm going to be focusing on how we can increase this efficiency. And this is because this has been really a room for growth in lighting. So lighting consumes a large chunk of electricity that we use worldwide, and this has lent room for lots of innovation. We've seen, like incandescent bulbs, fluorescent light, and most recently LEDs really taking over the lighting industry. And it's because it's such a ubiquitous technology that a lot of us use in our daily lives. But at the same time, there's still over 1 billion people worldwide who are without reliable lighting, and this is where semiconductors and nanotechnology have really been able to make strides in making energy more accessible. So solid state lighting, again, LEDs, as you might commonly know them, have really pushed for efficiency and savings. This down here is a chart from the Department of Energy that shows the projections that we've actually been able to accomplish now by 2020, but in the next 10 years, actually over double the amount that we've already been able to save, which is really impressive and leaves a lot of room for opportunity for researchers to take. I think like chemistry is typically, it's like bench chemists making like small batches of material, but really innovate and scale these technologies to make them accessible to as many people as possible. And that's because these technologies operate on a system that minimizes the amount of waste heat that we get out. And that's because what we're doing is eliminating that mechanical work that's typically required to generate energy. So if you're burning fossil fuels, what we're taking is like taking oil, burning it, using that heat to generate steam that spins a turbine, and that process then generates the electricity that we use in our homes. With solid states, though, it's a diode. So what that means, so diode like being two, so it has like these two opposite ends that drives the electricity going from one way to the other. So it eliminates that need to have mechanical work that drives the processes in a much more efficient way. So you could think about it as taking like one step instead of taking like 3 steps to get your electricity out. And this works by simply pumping electricity in. So, you turn your light switch on. This pumps electrons into what's called an excited state, or their conduction band. And when this happens, I think like a lot of people, like when you're like feeling anxious, you're a little bit excited, you want to relax. So what happens is you release all that energy, and that energy comes out in the form of light. And that's great, right? So what's really nice about this is that depending on how much energy you're consuming to do that, so depending on the distance or the band gap between these two materials, we actually get different colors of light. So on the picture on the left here, I have like red, green, and blue. These all come from the same material, but by somehow tuning that band gap, we can actually tune the wavelengths of light from going, down to like infrared or heat cameras, which are really popular right now for like driverless, like automated driving, all the way to X-rays for cancer detection, or as we use them in our daily lives, visible spectrum. So this has been really, really exciting, because what it means is from one material or from a small class of materials with similar properties, we can then tune the amount of applications that we can use solid state lighting for. And ideally, these would come from materials that themselves are recyclable, easily extracted, and sustainable in their material life cycle themselves. Of course, this isn't without its own issues. So LEDs have their own inherent inefficiencies built into them. And this comes from the fact that I think, like, just like in the business of daily life, when you want to get from one place to the other, the path isn't always so straightforward. There are obstacles that get in the way. In LEDs, these are called trap states. So rather than jumping from conduction band to valence band, what we get is a jump down from the electron, just the conduction band to the trap state. And that's kind of a loss of energy on the way there. So what can we actually do about this, right? So this is where nanotechnology really comes into play. In the previous slides, I was talking pretty broadly about materials and chemistry, but what's inherent there is that I had mentioned that these are material defects, which means that we have to kind of take a step back and think about, okay, what materials are we actually synthesizing, and how can we engineer them to minimize those defects? And surprisingly, it's a lot like making soup in your kitchen. So right here we have, this doesn't look like this in real life. It's like a much kind of intimidating glassware setup in the lab, but this is like a typical inorganic chemistry setup called a Schlenkline, where we have a broth that we heat up to a certain temperature, and over a certain time, we actually nucleate particles. If you've made rock candy or if you've made stew, it's a similar process where heating the solution up to a certain temperature generates particles, and you get this like nice chemical soup. Or in this case, what actually comes out are these small, tiny little nanoparticles. This is an image of them underneath the microscope. And this is really nice because it's like playing Legos at this point. Rather than having these big bulky materials that we have to figure out, okay, is this one good for blue? Is this one good for red? We can actually start with all these different salts. And just by, just like the way you would in the kitchen, maneuvering time, maneuvering the concentration of your ingredients, maneuvering the temperature, you get a precise control over the shape and size of your particles. And the reason these are called quantum is because what happens is when you get to this small size, you actually lose that band structure that I had shown in the last slide, where you had like jump from top to bottom. And what you actually get is these bands become separate states that you can actually start to see. So the smaller you get, the more you can actually like see on the level of what electrons are actually doing. So you're kind of seeing eye to eye with what's going on in atomic level, which provides us a lot more control than you can get in just like bulk materials. Which means that if we go back to the graph that I had shown previously, where we showed this like spectrum of light that we can get, we can add one more parameter, which is super exciting, which is just by having one material and tuning its particle size, you can get a whole spectrum of colors, which is really nice. So you could just like grow something for longer, for example, it's a little bit bigger. Now I have a red LED. If I grow it for maybe a second, I get a blue LED. And this is really exciting because this are, as you can see, are in solution, which makes them, just like you have like ink in your printer, you can actually print them on like large substrates. And this is how, if you've seen lately, Samsung has produced these like QLED TVs. This is the same technology that they're using. And this has been making strides in other applications as well. So a big one is catalysis, dealing with carbon capture, which has been of interest a lot lately in political sphere as well. Thermal materials, so a fun application is like actually generating invisibility cloaks on the same idea of absorbing light or infrared waves or heat, and then using that as a mask to cover up cars or yourself. And then also in biology, so some people are interested in certain materials for diagnostics, if you're able to like shine a fluorescent light after like taking an indicator or just like mapping out tumor images, which is really exciting. And then, so that's kind of where the research is going. So the nanotechnology part of it is kind of like you start really small, but the implications can be very, very large. I know we're running briefly on time, so I'll just briefly go over why this work is so important to me. And Michael had mentioned it a bit too, but outside of the lab and Noel as well, I've been working on how we can actually support sustainable development of these new technologies. And really it comes down to just like one thing that I think everyone can do, and that's just to get organized within their communities. And this for me has meant working with March for Science, which for those of you who are unfamiliar, was a reactionary moment, kind of in the same vein as the Women's March, to lack of funding for science, lack of respect for government scientists, but has really transformed into a network of volunteers all over the world who are dedicated to advocating for STEM and STEM advocacy. In our group, we are working a lot with community organizations to uplift communities around issues such as climate and healthcare, especially since those are the issues that New Yorkers have expressed a lot of interest in working on. I'm happy to talk more about that, particularly our abolition climate justice work. But essentially what our role is here is to really see how we can increase representation in science by having these conversations with people who might not see themselves in science, but to really flip the narrative about how these technologies get implemented. So if we're talking about like, building a solar cell farm on on Rikers Island, which is the jail that's going to close in New York City, who does that energy like actually belong to? Like, where should it be placed? Who has access to this energy? So thinking about, again, I think in the same vein of these life cycles of where the technology goes. I think what this comes down to too is just like seeing your place. I think like as someone who hasn't wasn't very politically active before getting involved with March for Science, it was very intimidating to kind of jump for it and go for it. This is a graphic from Sunrise Movement that shows different ways that people can get involved. Something that I thought of doing was mapping out how my PhD skills and everyone's skills as scientists can really lend themselves to this space. So for comms leads, we're here today sharing our knowledge, but this is, making figures, writing papers, giving talks. As hosts, we have these PowerPoint parties, we coordinate our lab meetings. We're always organizing in teams, right? As Carla had said, and others had said too, like this work is never done alone. And then as action lead, maybe we're not running campaigns politically, but we are pioneering work in our fields. That takes a lot of courage, a lot of organization that maybe doesn't seem exactly like organizing for climate, but does require some kind of, I think, like gut to do so, And then again, recruitment lead, managing collaborators, all of that. And then to JP's point, at the end of the day, it's about storytelling with your data, really using that data to uplift your work in your communities. So that said, I have a list here of community organizations like to consider and to look at. I'm happy to talk more about this later on too, or you can reach out to me. I just want to say thank you, and I'm happy to take any questions.
Thank you, Ingrid. If anyone has any questions, please drop that in the chat box. I'll start us off. I kind of have two. One that is super fun, and I got distracted when you showed the invisibility cloak. But my first question is, what is the mood of, I don't know, the movement toward renewable energy right now in societies, not just in the United States, but in like developing countries? And how feasible, or like when do you see the time when that will be the norm?
It's going to be the norm as soon as I think as more developed quote unquote countries like the US, European countries take the lead, because unfortunately that's just where the capital is. Like we are the most, and these countries are the biggest producers of carbon emissions. Like that's just how it is. So developing countries unfortunately lack the infrastructure a lot of times to make that jump to renewables, but they're also suffering the most from climate disasters like live. So I think it's going to take that leadership to really do. One thing that's definitely going to need to be done is, if you're familiar with COP, which is the like UN climate summit that happens, I believe every year. Every year. I think it just canceled last year because of COVID. Is they actually don't even consider the IPCC report on climate change, which is like the official like statement of rather doom that says, in 12 or so years, climate crisis is going to be irreversible. That documentation is actually not even considered during negotiations. So it's kind of like, we have our scientists like looking at this, but we're not going to talk about it when it comes to solutions. So that's something that needs to be addressed.
Okay, and then I think I'll ask this question that popped, or a comment in the chat box related to that. renewable is great, but it can't be at the expense of indigenous people's resources. So I guess just like the conflict between or imposing or promoting that versus the technology that's needed to get that up and running.
Yeah, I 100% agree. And this is part of the abolition climate justice work that Michael had mentioned. So a bit of context is that Renewable Rikers is this plan to take Rikers Island in New York City, which is a jail that has just a terrible, terrible reputation, close it down and turn it into a renewable energy farm, which is really great for a lot of people who are living in that area because it would allow us to close all of these peaker plants in the Bronx that have caused higher rates of asthma among people there. But at the same time, there's two things. One is that when we close Rikers, the plan is actually to open other jails. So it actually doesn't deal with this issue of managing incarceration at all. The second is that who does this land at Rikers actually belong to? Like right now, the plan is to give it over to the EPA, then turn it into a solar farm, but the land was actually never ours to begin with. And there are still communities that are just like waiting to see what will happen and advocating to give that land back. So that's something that definitely needs to be part of the conversation.
And then I'll take a couple more questions. So how do you think, like what's the ethical way to scale up technologies like these in less developed countries that may not have access?
Yeah, I don't think there's a one size fits all. I think it needs to be a partnership. I think back to the first and second comment and question that you had mentioned, where local, and I think it goes back to Kathleen's point too about like really valuing local knowledge, because these are the people that know the areas that they live in and what's happening, right? So I think if we're thinking about solar energy, if we're thinking about wind, it really needs to be that kind of maybe turning a dial up and down to what community needs are, rather than having this one size fits all.
Yeah, totally agree. So context. And then this question by Kelvin, does your research also look to examples in nature like bioluminescence for materials or chemicals that are more efficient for producing light?
I personally do not, but there are folks who do. So there's a whole field of nanophotonics that model their work, just like trying to match the optics of nature.
Okay, and then he had another question, but I think we can actually ask that and open it up to the bigger group, and I'll have Yasmin take over. But Calvin's last question was related to the earlier question, was that how do you recommend your technology be used to uplift communities through jobs and environmental justice?
Yeah, definitely. I think the most number one thing, so if you're a bit unfamiliar with like scientific research, particularly in chemistry, the way the funding goes is like these programs are usually funded by big agencies like DOD, DOE that are usually looking at these things for military applications, which means that they can afford to look at materials that are very expensive either themselves to extract or to process, or materials that require a lot of work to process, like gallium nitride, getting a bit in the weeds, is a LED material that is amazing, but you need to process it on sapphire. And that's just not going to work if we want to get lighting available to everyone. So it really needs to be done, that kind of like material life cycle assessment of looking at what elements we're even looking at to create these materials. And then when it comes to jobs, I think to Yasmin's point too, like this, like just like demystifying and knocking down these doors in science that exist for people who might not see science as accessible to them is really important. And I think that's something that requires a bigger effort from like education, government, a lot of fronts to really tackle.
All right, thank you, Ingrid. Another round of applause for our final speaker. And I'll have Yasmin take over to facilitate the discussion.
Yeah, everybody, thank you so much for your incredible presentations. I feel like my brain is full. I'm sure everyone else feels that way. I would like to kick off our discussion with a more broad question. So we know science work is hard. It's difficult. I'm sure that in order to give your presentations today, you've had to go through like months and years of, grueling work. So the question I have for you is, what are the biggest obstacles you are facing in your work now? And not necessarily scientific obstacles, although could be that too, but also like structural, societal, you know, what is getting in your way, and how are you getting past them? Anyone want to jump in? I know it's a big question.
I could jump in.
Yes, please.
I think one dilemma I have, especially it's been really highlighted during the pandemic, is how I see my science being very related to what's happening right now. Because sometimes when I'm in the lab, it's really like all things are technical. Like you need to look at the cells, but it's hard to grapple like solving these very technical questions when everything in the world is crumbling down. Of like, yeah, the pandemic is here, and there's a lot of social injustices happening outside. So I I had to grapple like the balance of being like the scientist in the lab, but also like what is my role really in like in the society as a scientist trained to critically analyze things and being in still a problematic institution in itself still, right? And so how I actually grapple with that is I um, talk to a lot of AFSA members, and we're very involved with like different advocacies and campaigns, and we have like support groups as well. And we get to talk about all these matters that actually, you know, like that are things that you can't just find inside the lab, and it made you more real as a scientist, I would say, and it gives you like a lot of opportunities to really take action against the social injustices that you see. So yeah, being in a community, I guess.
That's great. Yeah, I'm sure being in a lab, you get a sense of tunnel vision and you forget what all of your other amazing skills. So I think community is important to help you remember that. Does anyone else have thoughts? Want to share what they're struggling with and how they're getting past it?
I can go. You know, and this comes again from my own disciplinary footing. I would say that the crisis in the humanities is not new. It's something that we know and has been on the tip of our tongues for decades. It is incredibly felt right now. I think, especially in this moment with the pandemic, it is getting harder to even think about humanistic engagement when in fact we're confronted with material problems that are crumbling right before us. I come from a campus, both from my PhD and from teaching, where we are becoming a much more STEM-oriented location, right, for development, and this is great. I also think that there are opportunities still for the humanities to engage in that dialogue with STEM, especially if you go into something like science and technology studies, which allows for a bridge between those fields. And for people like me who really believe that there's a lot to be gained from both a partnership with the humanities and with STEM. But I would say that right now, as we're kind of being faced with not only economic constraints, but economic demands, it's getting harder and harder to really show how the humanities still has a place in scientific thought.
Absolutely. I could not agree more that these approaches are necessary. This relates to another question that I have, but I wanted to see if anyone else, Ingrid or JP, wanted to share any thoughts. If not, I'll move on to my next question.
Yeah, I just want to say that both what Kathleen and Carla said really resonates, particularly, I guess, like in a flipped institution where I'm at a STEM institution. And like, I think we've seen in the last year how when we don't have these lessons of empathy, like these things, like everything around us just like breaks down, whether it's like COVID or the climate crisis, and thinking about that context of like where our work fits in, even if it's like the accessibility that I provide and empathy that I provide for like students I TA to like the lab work that I'm actually doing. It is a difficult balance to do, but having a good support system, I think, is always key.
I think mine is related to this idea that climate change, it's happening, but actually its effects might, it's not quite felt in many parts of the world. In some parts, yes, but it's hard to wrap your mind around it when you're seeing other much more pressing and immediate issues. So then, thinking about your skills, or my skills as a molecular biologist, as a microbiologist, and thinking about, well, I could have actually devoted that and directed that towards helping with some aspect of this pandemic, as opposed to doing more environmental work. But I guess I have to always think about the long-term, some of the long-term issues that we're going to face in the future. So yeah, it's all related to all of that.
Yeah, in my own work as a science journalist, I find that communicating the sort of long-term view that scientists often take is really difficult. People expect science to deliver quick solutions all the time, when it's like, no, we're thinking like years ahead. But this brings me back to a question I wanted to ask all of you. We have seen, obviously, in the past year, a really horrible anti-science movement in this country and in the US and around the world. And I'm curious how each of you address it when you encounter it, where you encounter it. Maybe it's even like in your own families. I've certainly experienced that. And I'm curious to know, how do you bring empathy into that conversation? How do you have the patience to have these conversations when it's so frustrating?
I'd be really happy to jump in on this, in part because this comes up, especially again, if you're in the humanities, and you might be confronted with the question, well, you just showed that science is an amalgam of cultures and decisions. It's very human. So it leaves up the room for error, right? So how should we invest any trust in science if you've just shown us a really messy history behind it? And I actually think a way to go around that is by understanding process and the work of science. And this is really common in the philosophy of science, where the work that we're supposed to do as historians is also show the process of reaching the empirical claims that we do when we do reach them. And like you were saying, Yasmin, this takes time. This is not a silver bullet for society. And we have to really ensure that people understand this. And I think that's part of the problem that we're seeing with COVID right now, and the denial around its severity, because we're seeing science in action. We are seeing mistakes made, mistakes rectified, and we're moving forward. And I think it's blowing people's mind to see this process real time, when in fact we've been confronted with history textbooks that show the clean process of scientific development, when we in fact know that it's never looked that linear. And I think this is the role, again, of us historians and us humanists, to make sure that that process be understood in a much more realistic way.
I could not agree more. Having covered the pandemic for the past year, I've been reporting in real time on the research that's coming out. And so often I look back like a month ago and I'm like, oh, that paper was wrong. But, I feel like the world's getting a crash course in how science actually works right now, with all of its imperfections. So that to me is an important thing for people to learn. But I'm curious to know what everyone else what lessons you could teach us about science communication.
I think I just have one quick point, and it's about the public's perception of what uncertainty is, and how scientists are often, they're not quick to make bold claims. And I think a lot of people perceive this as lack of information, as kind of the weakness of science, but actually it is a strength of science, in that people are, I think it shows cautiousness and carefulness in interpreting the data, especially as they come out, and without full data, without long-term data in many respects, it's very difficult to become confident and to make bold claims. So I think part of my task is to tell others that uncertainty and often this wishy-washiness way of speaking. It's just so that people can be certain when they claim a fact as a fact.
Yeah, I think the only thing I'd add to that is that's where storytelling really becomes important. I think particularly in climate, I've met folks who just really just don't believe in it at all. And I think when at that point it does become frustrating, right, to have empathy with someone who just is like on a different like wavelength from me completely. But there is something that we can come to grounds on, hopefully eventually, whether that comes to, you know, they don't understand climate. Maybe they don't see like emissions as the issue, but they do see, if they're a farmer, like what's happened to their crops year after year. Or in the city, even in New York, we've seen like heat waves year after year, and that's causing like more and more damage to our communities. I think making those connections to what's really happening on the ground and then turning that into a conversation about data really helps to flip the story. So I think, yeah, when we see like, even like as a scientist coming into a field that's like not my own, when I see all this data, it's just like, okay, like what's going on? But taking that step first to really talk to someone about what's going on with them can be a bridge to build.
Ingrid, Carla, please.
Oh, yes.
Oh, I just wanted to agree wholeheartedly with Ingrid that you have to lead with emotion sometimes, because people get freaked out by science. We know this. And so I think to appeal to their fears or what they hope for, I think that's a really important way into having these conversations with people. Carla, please.
Yeah, I'm also agreeing to what everyone said. But this is really a point of reflection on me, on especially with what happened with COVID, of like how scientists could be really secluded sometimes from the community, and how we could be really elitist at some point. That whenever I read information personally, I would always say like I understand it, but I don't get how other people are not able to understand it. So I'm not able to really interrogate where I'm coming from. And I think as scientists, we really have the responsibility to actually integrate with the communities, just like what Ingrid said, and really know what's happening on the ground and how and why they're not understanding the concepts, and what are the better ways that we could translate the scientific terms and concepts into something that is digestible to them and they would understand in layman's terms. And another way, I guess, is, and I think it was highlighted during the pandemic, AFSA has been doing a lot of information campaign. And one factor that we really had to look into is how Filipinx have different languages. And even that, like how we use language to actually inform people, because there's Bisaya, there's Ilocano, and not everyone actually understands English. And so like, how do we approach that in a way that's really rooted to where their communities, where these communities are, and really learning like where are they coming from. And aside from that, really like how we also need to think how leaders actually understand science and follow science, because you know, like what happened in the US and in the Philippines, like if the leaders themselves don't believe in the science, like bleach as disinfectant or gas as, you know, like something you could drink or some things like that, you also need to really count that into the picture. So I would say like scientists really have a very great responsibility to play, especially in instances like this, and not really just to be secluded in their workspaces, but to go out in the community and know their struggles and how to better relate to them.
I could not agree more. And it really sounds like everyone who's presented today has a real concern for engaging with communities, and you know, you've all been doing that. So I'd like to bring up Noelle's question here. Having incorporated advocacy and justice in your work, not divorced from your science work, how do you feel like your work has changed as a result?
I feel like for me, it's definitely informed how maybe not the technical parts of my project so much, but definitely how I interact with people at my institution, whether it's my mentors and also administrators. I feel like as a younger grad student, before I was really attuned to all the issues going on, I think it's kind of like, yeah, like kind of just like getting disillusioned as things were going. And then seeing as my advocacy work in the community and my institution and my research as two separate places of dealing with. But as I started to work more with the community, I started to really realize, particularly within science, right, we have issues, related to like work-life balance, just as graduate students, and dealing with all the issues that get compounded depending on your identity, how I can use my area of expertise and also the power that I might have, at least a grad student to a student that I'm mentoring, an undergraduate student or high school student, or on a colleague level with a faculty member, how I can really use that to inform change within my institution. That's been something I think is like ongoing though throughout.
It's awesome to hear that you're doing that. I feel like the more of that, the better. Anyone else?
I think for me, it shows up in my extracurriculars and how much time I devote to things not directly related to the work that I do, i.e. carrying out the experiment, analyzing the data, publishing the paper. It's being a lot more mindful about how to communicate the work that I do in a way that a lot of people understand, if not, hopefully all people can understand. And it's also thinking about prioritizing time, deliberately allocating time for things like this, for engagements such as Skype a Scientist, where you can chat with classrooms worldwide. And I think that is probably how it's mostly affected my work, and it's in terms of the allocation of time and intentionality.
We have a question specifically for you, JP, but I'm gonna hold real quick to see if anyone else has something else they'd like to share.
I guess in my end, really like looking into the health sector and how, like there are available data on like genetic sequences of patients and stuff like that. But how is it even really, like what are, what are the classifications of these patients and who are really able to access the health facilities in the hospitals? And that's something that I've been like interrogating a lot in the lab, of like the accessibility of health even, and when this drug even translates to the clinic, like which like demographics is it really designed to? Is it like inclusive to everyone? But also another aspect is really like coming from the Philippines, to have really experienced the science in there. Like there's a lot of privileges with me coming in here and really doing like very advanced technology, advanced research, because in the Philippines there's a lot of, like the funding is not even there, and people had to grapple like having no salaries or research assistants. And so like aside from this, like coming from the privilege of being able to do this work, is really looking at how are we able to impact our communities and really expose the systems that are not right, and really try to change that in a way that we could with the collective and community that we have.
And I'll just add really quickly, and it's a big plug for the Science and Justice Research Center that we have at UC Santa Cruz. It's, in my opinion, one of the most amazing STS sites that really has justice as a core foundational tenet. We are revamping curriculum to make sure that justice is always a part of science studies discussions inside the classroom. And I think there is always a place for local knowledges, ones that maybe fall outside of the scientific ambit, that still need to be preserved and upheld in particular ways, especially because when we think about the scientific community, it is a tiny fraction of this 7 billion plus world. And we have to remember that those local knowledges, in whatever iteration they take, still deserves their place in the sun. And so I think the Science and Justice Research Center really does take that as a core value too.
Completely agree. In my own work editing this website, Future Human, I had to insist on a big change this year with my staff. We were covering climate change and climate justice as sort of two separate verticals on the website. But this year I sat down with my team and I was like, it's not separate anymore. Climate change coverage is climate justice coverage. And I was like, all the other stuff we do, biotech, genetics, all this stuff, it's all justice. It's not about finding a justice angle. We have to include it. We have to look through that lens for everything. So Dr. Gutierrez, I'm going to hit you up about that center. That sounds awesome. Oh, perfect. There we go. Okay, I want to get to a specific question that sounds like could be really fun. This is for JP. Tell us about your experience on the ship. What's that like?
I'm really glad that you asked, because it's rare that I meet another Filipino oceanographer. So I think this is sort of like a question that a lot of people ask. But in general, what you see, or what you saw in those photos, is that there's a lot of hard work that goes into it. And I think, there's some cultural differences depending on whether you're on a German research vessel or an American research vessel. The US has its own fleet of research vessels, but in general, it's usually around the clock work. And that's more so when these cruises are not as long as, let's say, Mosaic. So Mosaic, we had on average about 2-month-long segments out on the ship in the Arctic. But for other research cruises, they could be anywhere from as short as 5 days to a few weeks. And that is very, very intensive 24-hour work and operations. When we're not deploying equipment to collect water, we're carrying out experiments. The really special thing about this Mosaic expeditions, particularly leg 4, is we began the leg 4 on, I think it was the beginning of June. And that's in the middle of the pandemic. And as we all know from the news, right, ships are sort of these petri dishes of infection. And so there was a lot of scare surrounding getting on a ship, and especially when you're out in the Arctic where the nearest land is hundreds of kilometers away, and medical evacuation via helicopter could respond, and but they won't do so for many hours, if not days. There was a real fear for a lot of people, and we saw this. It affected us in part based on the composition of the team. So whereas usually you'd have very senior people going out to these expeditions, actually there was a huge shift such that we had much more PhD student and postdoc representation and a few senior scientists. What I didn't talk about also was the 3-week-long quarantine that we had to do beforehand, and 1 week of that was actually solitary confinement in a hotel room where they fed us through the door, and then once we passed 3 COVID tests after 2 weeks, then we were finally allowed to get on the ship. But of course, there was still a lot of uncertainty, and part of that was, well, if situations worsen and the next team that's supposed to relieve us, the last team to get on board, don't make it to the Arctic, then we're then going to have to carry out the rest of the leg. So then we'd be stuck there in total about 4 to 5 months, which is twice what we would have signed up for. But I think after getting through all of that, what it generated was a sense of accomplishment for the team and readiness to tackle pretty much whatever, right? You'd gone through this quarantine. There's a lot of fear, but I think there was also a lot of hope and people really pursued the opportunity, and the team worked very well together. And the captain of the ship, who has been a captain for the past few decades, commented on how well the team really worked together, that he hadn't quite seen in either previous legs of Mosaic or previous research cruises on Polar Stern. So I think it made for a really positive dynamic where everyone just really pursued any sort of opportunity to get their work done. So yeah, it was a very positive experience.
I think you have to have a positive attitude to make like 2+ months in isolation, you know, to seem such an encouraging thing. So great. I'm glad you did not have to stay super long. Okay, we have one more, time for one more question because we are running low on time. This one is for Dr. Gutierrez. Tell us about your dad. This photo that you showed us, how does his work factor into your research? Or does his work factor into your research? And if so, how do you approach studying a family member as a historical actor?
Oh boy, I was just messaging Michael about this. I was like, oh my God, it's been so colorful. I will write you separately. And so I'll just talk about it here. Thank you for that question and really bringing him into the fold. My dad lives with me now in Oakland, California. And so it's been a journey for us, especially during lockdown. I come at this in two ways. The first, in terms of research, traveling with kin, and then of course, having him as an intellectual interlocutor. I brought my dad through all of my historical research in the Philippines. It was exhausting. It was fun. It was strange having my dad fall asleep in archives while I was there, kind of looking over work, and it was transformative. I think there were places I got to see that would have not been accessible to me if it weren't for him and his presence, and the questions he really got me to think about as a person who has practiced botany for 50+ years. And so that was foundational. And I think there actually needs to be more written on heritage scholars like us who study the Philippines, for instance, who incorporate our families in the research process. So look out maybe for a panel discussion at some point. And the second comes with him as an intellectual interlocutor. And I say this a lot, my work are love letters to him. And I don't think I would have a lot of purpose in writing about the history of botany if I weren't really thinking about the trajectory he went through as a scientist, especially under Marcos, and leaving in 1985, in part because of the political repression then. And so I have written about my dad. I've incorporated him into my writing unapologetically. I do consider him a primary source for particular vantage points in history, but I don't not complicate it. And I don't write a heroic narrative about him either. I refuse to do that when it comes to the history of science. And at the same time, I think it's stories like his that really need to be uplifted because historians of science in the Philippines are really, really small. This is a field that we really hope is going to continue to build. This is what Paul Michael Atienza and I are trying to build with the STS journal and hopefully collaborations moving forward with all you all on the panel. And I think there is a place again for heritage scholars who recognize that there's a duty involved with how we uplift our family stories. And I think there's still a careful way of doing that, even when it comes to historical writing. It's not easy, but it's possible. Oh, and I should just say, so he got his PhD at the University of Santo Tomas. So he's a UST grad, but he was also a Fulbright scholar to Harvard back in the 60s during some pretty difficult times. And he has his own stories to share about that as a Filipino scientist trying to make his way in civil rights era United States. Again, a wonderful man.
Dr. Gutierrez, thank you for sharing that, that story. That it just sounds like such a journey the two of you have been on together. Oh, Noel says, let's have him in our next panel. I completely agree. Why not?
He probably invented the Filipinx PowerPoint party. He was probably doing this in the 60s.
Just like acetate slides. Okay, unfortunately, I think we are out of time for more questions. I'm sure everyone else has so many more, but like Dr. Gutierrez said, I hope we can all find a way to collaborate and keep this conversation going and share ideas for how to communicate and get involved with our own communities and share science with everyone else. I'm going to turn it back to Noelle and Michael.
Thank you so much to all of our presenters. I feel like I learn a lot at most of at all the PowerPoint parties, but I feel like this one I got taken to so many places that I had never just got to learn about stuff that I would never have learned about before, and so this was so exciting. So thank you to Kathleen, Ingrid, Carla, JP, Yasmin, and yeah, we'll see you all soon. Noelle, do you have anything to add?
Just echoing everything you said, and I mean, even as Filipino academics, it's so gratifying to find out about this community of AFSA and all the work that everyone's doing. And oh, Michael, can you add the Venmo account again? I think that's your Venmo. But thank you everyone for logging in this Saturday afternoon, or I mean, wherever John Paul is in Denmark. Oh, actually, it's almost midnight there. So thank you times 10 to you. And yeah, hopefully we see you in our next event, and that we can continue to collaborate and do science and do community work and advocacy, all in the name of Filipinx and promoting our just our goals and objectives. All right, I think that's it for tonight. If nothing else, we'll say our goodbye. We'll upload this recording in YouTube. We have our channel, and Michael will send out an email with a link and also follow-up questions and requests, if any.
And thank you all to everyone who donated. We got a bunch of donations, and so yeah, I'm sure AFSA and the March for Science are super excited about it.
Thank you, everyone.
Thanks, everyone.
Thank you.